There's a universe hidden from our naked eye, a world teeming with life too small to see. This is the microscopic world. Our journey into this realm began centuries ago, with simple lenses that magnified tiny objects. These early microscopes opened our eyes to a universe within. Today, powerful microscopes act as our spaceships, revealing the intricate details of cells, bacteria, and even viruses. These aren't just tools for observation. They are instruments of discovery. They allow us to understand the fundamental building blocks of life. But not all microscopes are created equal. Each type uses different techniques to illuminate the microscopic world. Each has its strengths, revealing different aspects of this unseen universe. Let's embark on a journey to explore these special microscopes and the profound impact they have on our understanding of the cosmos within. Imagine a starry night sky. The darkness provides the perfect backdrop to see the faint glow of distant stars. This is the principle behind the dark ground microscope. Instead of illuminating the entire specimen like a traditional microscope, the dark ground microscope uses a special condenser. This condenser blocks direct light from reaching the objective lens. What we see is light scattered by the specimen itself. The result is striking. The specimen appears bright against a dark background, revealing incredible contrast and detail. This makes it ideal for observing living and unstained specimens. We can see delicate structures like bacterial flagella and internal organelles in action. The dark ground microscope is a powerful tool for studying living organisms. It allows us to observe their behavior and interactions in real time. This is crucial for fields like microbiology and cell biology. Imagine painting different parts of a cell with light, each color revealing a specific structure or molecule. This is the essence of fluorescence microscopy. This technique relies on special dyes called fluorochromes. These dyes absorb light at one wavelength and emit it at another, longer wavelength. When illuminated with specific wavelengths of light, the fluorochromes attached to the specimen glow. This allows us to visualize specific components within cells, like proteins, DNA, and even specific molecules within these structures. We can track their movement, interactions, and changes over time. Fluorescence microscopy has revolutionized biological research. It allows us to study cellular processes in unprecedented detail. We can track the spread of diseases, study the effects of drugs on cells, and even map the intricate connections in the brain. Living cells are transparent and mostly colorless under a traditional microscope. This makes it difficult to observe their internal structures and processes. Enter the phase contrast microscope. This ingenious invention exploits the subtle differences in the way light travels through different parts of a cell. These differences, called phase shifts, are converted into variations in brightness. The result is a high-contrast image of living, unstained cells. We can see organelles, nuclei, and even the delicate dance of cell division in real time. The phase contrast microscope was a game-changer for cell biology. It allowed scientists to study living cells in their natural state, opening up a whole new world of understanding about life's fundamental unit. Light microscopes have limitations. They can only magnify objects up to a certain point. To see even smaller structures, we need to turn to a different kind of illumination: electrons. Electrons have a much shorter wavelength than light. This allows electron microscopes to achieve far greater resolution and magnification than light microscopes. We can now visualize objects thousands of times smaller than a human hair. There are two main types of electron microscopes: transmission electron microscopes, or TM, and scanning electron microscopes, or SEM. Each provides a unique perspective on the nanoscale world. Electron microscopy has revolutionized our understanding of the ultra-small. It has revealed the intricate machinery within cells, the structure of viruses, and even the arrangement of atoms in materials.